All right, we are here to chit-chat about glycolysis. I used to hate this, but now it's like an old friend. So the first thing is I can describe the process of glycolysis, and we're going to go into a lot of detail, but just very briefly, we can break this word into two parts. Glyc, which means sugar, okay? And so that's why you hear like glycogen, glucose. Gluc is another prefix for sugar. And lysis, which means to break or to burst. And so glycolysis is literally the breaking of sugar. We're going to take a 6-carbon glucose and we're going to break it into 2 3-carbon molecules, okay? So I can describe the process of glycolysis. It is the breaking of sugar. I can tell where it occurs. It occurs in the cytosol. We are going to talk about the fact that it is anaerobic. And it is going to make some ATP, and it's going to make it by substrate level phosphorylation. Okay, and if you're not sure what that is, go back and watch the foundations of cellular respiration. I don't remember, something like that video, and it'll go over substrate level phosphorylation. All right, so I can tell where it occurs. It occurs in the cytosol. So it is the one part of cellular respiration that does not happen in the mitochondria. And so organisms that do fermentation, which we'll talk about that next week, or this coming up week, or this week, depends on when you're watching this video, they can do glycolysis followed by fermentation without a mitochondria because neither one of those processes require mitochondria. Let's see, I can list the reactants of glycolysis and the products. So what we're going to do, oops, I'm getting ahead of myself there, don't need an R, it's not pyruvate, it's pyruvate. Is we're just going to draw it out and then we're going to list the reactants and the products at the bottom. All right, so glucose is a 6-carbon molecule, C6H12O6. Okay, it's a carbohydrate. And we're going to simply break it into 2 pyruvate, and pyruvates are 3-carbon. Okay, so all of your carbons are accounted for. The reality of this is that this is actually like 10 steps, and we are showing an overview. So just know, while I'm giving you a lot of details, there's a lot more details that I'm not giving you to. All right, so we have to use a few ATP to get started. So we're going to burn up 2 ATP, break off that 3rd phosphate, release that energy to get going. But in the process, we're going to make 4 ATP. So that means we have a net gain of 2 ATP. Okay. Also, anytime you break a bond, you are going to release electrons, and they will be carrying energy. So you'll hear me say it a lot of times, you're going to release energy in the form of electrons. So what's going to happen is when we break that 6-carbon glucose, I got to get my directions okay, that 6-carbon glucose into 2 3-carbons, we've broken that bond between these 2 carbons, okay? When we break that bond, some electrons are released and they take energy with them. So we're losing electrons off of glucose. They are going to go to a molecule called NAD+, there's actually 2 of them, and it's going to convert that molecule from NAD+ to NADH, okay? So NAD+ is obviously a positive molecule. We're going to add another positive, a hydrogen ion, and 2 electrons, and that's going to give us 2 positives, 2 negatives, and that's going to give us our NADH. So every NAD+ can carry 2 electrons, okay? And so here we have 2 NAD+, that means we're going to be carrying 4 electrons out of the glucose. So since glucose lost electrons, it is oxidized, and since this guy gained them, it is reduced. Okay, so just keeping an eye on those redox reactions as they happen. All right, so, and then we also lose a few waters. Now these waters are coming off, the hydrogens and the oxygens are coming off of this glucose. So this is why, even though this is anaerobic, you're seeing oxygen come off. That is not because the oxygen, it's not like oxygen is swinging in and picking up the hydrogens and leaving. This is not atmospheric oxygen. That's what it means to be aerobic. It uses atmospheric oxygen. This oxygen came off of this glucose, okay? So that's why it's anaerobic. As far as the reactants, let's make a little list here. Reactants is everything going in. So I've got some glucose going in. I've got my 2 ATP coming in. I got my 2 NAD pluses coming in. Okay? So those would be my reactants. And then my products, I have my 2 pyruvate. I have my 4 ATP, or you could say a net of 2. I have my 2 NADHs, and I have my water. And I think there's 2 waters. We don't really dwell on this water. A lot of times you'll see me draw this. I won't even include the water because it's not crucial to our understanding. So I'm not going to go stop the video and go look it up. I think there might be 2 waters. It might just be 1. So anyway, if it's 2, just know there's a 2 there. If not, then it's right. All right, so I can describe the process of glycolysis. It's that breaking of this sugar in half. I can tell where it occurs in the cytosol. I can tell what it means to say it's glycolysis. It does not need or use any atmospheric oxygen. I can list the reactants of glycolysis. I can list the products of glycolysis, and I can explain the movement of electrons in glycolysis. Those electrons are moving from glucose to NAD. One of the things you want to do as you go through these steps of cellular respiration is follow those electrons. It's all about redox. I mean, literally, it is all about redox, because redox is pulling those electrons off, and therefore the energy, and that's the whole job of respiration, is to release energy and put it into ATP. We break our food down to do that. So when we talk about the movement of electrons, you do want to be able to follow these electrons from glucose to NAD+, and then eventually to the electron transport chain and oxidative phosphorylation. So that is kind of glycolysis in a nutshell, an overview. That's not an overview. That's a pretty detailed picture. But I hope that helps you understand it. If not, watch it again a time or two. Draw it out multiple times. That helps a lot of students. Use different colors. You might want to, do your NAD pluses and NADHs in one color and your ATPs in a different. And as you're drawing all the steps, you kind of keep those colors consistent so that you really remember where we're producing those products. That helps a lot of students as well. So if you've got questions, if you need help or want me to explain it again, just let me know.